Hello guys, in this part I want to talk about glial cells and synapse. First, I will talk about synapse, and after talking about this part, you know what is the meaning of synapse, and you can describe the essential structural and functional differences between the chemical and electrical synapse. The synapse is the place where a neuron is functionally connected to either another neuron or an effector. The effector may be a muscle or gland. There are two types of synapse in human body, chemical and electrical synapse. Most synapses within the nervous system is the chemical one. In this picture, you can see the chemical and electrical synapse. In the chemical synapse, you see that we have a space between perisynaptic and postsynaptic cell. But in electrical synapse, the perisynaptic and postsynaptic connected together tightly. First, I will talk about the chemical synapse. In the chemical synapse, which is between two neurons, we have different parts. First, we have perisynaptic neuron. The perisynaptic neuron is the axon terminal of the first cell, which can produce signals. We have postsynaptic neuron, which can receive signals, and most of the times, the postsynaptic neuron can receive the signals through its dendrite. And we have synaptic cleft. Synaptic cleft is the small fluid-filled gap between two neurons. Now, I want to talk about these different parts for you. First, you need to know that the synapse may be between the axon of the perisynaptic neuron and any portion of the surface of a postsynaptic neuron, like dendrite, cell body, or axon. And sometimes the postsynaptic cell may be our muscle or our gland. Most commonly, a synapse is with a dendrite of the postsynaptic neuron. The synaptic knob of the perisynaptic neuron doesn't quite make contact with the postsynaptic neuron. The two neurons are separated by the extremely narrow fluid-filled gap, which we call it synaptic cleft. In this picture, you can see the axon of the perisynaptic neuron and postsynaptic neuron, and between them we have synaptic cleft. Transmission between the perisynaptic and postsynaptic neuron occurs when neurotransmitter molecule stored in the synaptic vesicle here are released in the synaptic cleft. Some of the neurotransmitters diffuse across the synaptic cleft, and some of them can attach to the receptor on the postsynaptic membrane. Because of that, we have a synaptic delay with release of neurotransmitter in the chemical synapse. The delay is the time between the neurotransmitter released from the perisynaptic neuron, diffused in the synaptic cleft, and then attached to the receptor on the postsynaptic membrane. This delay may be between 0.3 to 0.5 milliseconds. And you should pay attention that one polysynaptic neuron may and often does receive signal from more than one perisynaptic neuron at the same time. In this picture, which you have the animation in your own PowerPoint, you can see the perisynaptic neuron. Here we have the axon terminal, which is bulged part of the axon, and you can see the synaptic vesicles, which are full of neurotransmitter. These neurotransmitter can release in the synaptic cleft when the signals reach the end of our axon. When the neurotransmitters are released in the synaptic cleft, some of them are diffuse and some of them can attach to the receptor in the postsynaptic membrane, and they can make effect on the postsynaptic membrane. I told you that the postsynaptic membrane may be the membrane of the other neuron or maybe the membrane of the effectors like glands and muscles. The other type of synapse which we have in our body is called electrical synapse, and it's less common. An electrical synapse is composed of a perisynaptic neuron and postsynaptic neuron, which are attached together. Gap junctions, as you see in this picture, are located between the cells, and it can facilitate the flow of ions between the cells. The cells act as though they shared the plasma membrane. Thus, the electrical synapse passes between the cells with essentially no synaptic delay. Electrical synapses are located between the limited regions in our brain and also our eye. Now you can answer this question. What is the chemical synapse within the nervous system and how does it function? The next part is about Gilial cells or neurogilial cells. Gilial cells are sometimes referred to neuroglia. They are found between both the central nervous system and peripheral nervous system. The Gilial cells are both smaller than neuron and they are capable of producing new glial cells by doing mitotic division. Glial cells don't transmit electrical signals, but they assist neuron with their function. The glial cells cooperate to physically protect and help nourish neuron, as well as provide the organized, supporting scaffold around the nervous tissue. During development, glial cells can form the framework that guides young migrating neuron to their final destination. Recent evidence has shown that glial cells are critical for the normal function at neural synapse, both maintaining the anatomic structure of the synapse and modifying transmission that occur there. The glial cells far outnumber neuron. The nervous tissue of a young adult may contain 35 to 100 billion neurons and 100 billion to 1 trillion glial cells. Collectively, glial cells account for roughly half the volume of the nervous system. In this picture, you can see the different glial cells. This glial cell, let me make it bigger, is called astrocyte. This one is called microgiliocyte. Here you can see oligodendrocyte. So you see that we have different types of glial cells. Here we have the ependymal cells. And now I want to talk about each of them specifically. First, about the astrocyte. The astrocyte is a star-like shape due to projection from their surface. There are numerous cells in our central nervous system. They can connect both with neurons and blood capillaries. Astrocytes are the most abundant glial cell in the central nervous system, and they are over 90% of nervous tissue in some area of the brain. Astrocytes can protect, nourish, and support the neurons. Here you can see some of the functions which our astrocyte can do. One of the functions is help formation of the blood-brain barrier. Look at this picture. In this picture, you see that The ends of astrocyte process, which we call the perivascular feet, can wrap the blood vessels in our brain. These perivascular feet and the brain capillaries together can make a blood-brain barrier, or BBB. The BBB strictly controls movement of substances from exiting the blood and entering the nervous tissue in the brain. The blood-brain barrier can protect the delicate neuron of the brain from toxins, but at the same time allows needed nutrients to pass through. The other function of the astrocyte is regulation of tissue fluid composition. Astrocytes help maintain an optimal chemical composition of the interstitial fluid, the fluid which we have around the cells in the brain. For example, astrocyte can regulate potassium ion concentration by absorbing these ions to maintain a constant potassium ion concentration, which is very critical for the electrical activity of our neuron. The other function of the astrocyte is formation of a structural support. The cytoskeleton in the astrocyte can make the cell become strong enough to provide a structural framework to support and organize neuron inside the central nervous system. The astrocyte can assist neuronal development. Astrocyte can help directly on the development of the neuron in the fetal brain by secretion of different chemicals. These chemicals can regulate the formation of connection which we have between neurons. And the last function of these cells is occupy the space of dying neuron. When neuron damaged or die, the space formerly occupied is often filled by astrocytes that replicate through cell division. So this is about astrocyte, the most numerous one, which is the star shape, as you see in this picture. The next glial cell which we have in our brain and the spinal cord is called oligodendrocyte. Oligodendrocytes, as you see in this picture, are large cells. And as you see, they have few extensions. The extension of oligodendrocyte wrap around and insulate axon inside the central nervous system and form myelin sheath through a process which we call it myelination. This insulation allows for faster propagation of action potential along the axon. Here you can see the cell oligodendrocyte and the few cell extension which come out of the cell wrap around the axon to form myelin sheath. The next glial cell which we have in central nervous system is called microglia or microgiliocyte. They are typically small cells that have slender branches extending from the main portion of the cell. They represent the smallest percentage of central nervous system glial cell, with some estimate of their prevalence as low as 5%. They are classified as macrophages or phagocytic cells, and they are from the immune system. Microglial cells wander through the central nervous system and replicate in response to the infection. They protect the CNS against microorganisms and other potentially harmful substances by engulfing and destroying them through phagocytosis. Microglia also function in removing debris from dead or damaged nervous tissue that result from infection, inflammation, trauma, and brain tumors. And the last Gilial cell, which we have in the central nervous system, is called a Pandimal cell. The ependymal cell, as you see in this picture, are ciliated cells. They are simple cuboidal or simple columnar epithelium. They contain cilia and they line the internal cavities or ventricles which we have in the brain and the central canal in the spinal cord. These cells have a slender process that branch extensively to make contact with other glial cells in the surrounding nervous tissue. Ependymal cells and the nearby blood capillaries together can form a network which we call it choroid plexus. And the choroid plexus is important for making cerebrospinal fluid. This fluid is a clear liquid that bathes the external surface of the central nervous system and fills its internal cavities. The cilia we have for this ependymal cell help circulation of the CSF or cerebrospinal fluid. So the glial cells which we have in the central nervous system are listed here for you. We have astrocyte. The astrocyte can help formation of blood-brain barrier, regulation of interstitial fluid composition, provide structural support and organization to the central nervous system, assist with neuronal development, and replicate to occupy a space of dying neuron. The ependymal cell, as you see here, lines the ventricle of the brain and central canal of the spinal cord, and they assist in production and circulation of cerebrospinal fluid. These cells contain cilia, and the cilia can help circulation of CSF. These small cells are microgiliocyte or microgilial cells. They are phagocytic cell that moves through the central nervous system and protect the central nervous system by engulfing infectious agent and other potentially harmful substances. And the last cell, glial cell, which we have in the central nervous system is called oligodendrocyte. They can make myelin sheet and insulate the DNS axon, and it can allow faster action potential propagation along the axon in the central nervous system. Pay attention. All of these cells, which I mentioned here for you, are the glial cells which we have in our central nervous system. Now, the glial cells which we have in peripheral nervous system. Two types of glial cells are found in the peripheral nervous system. These specialized glial cells function in insulating neuron and include satellite cell and neuronemocite. First, I want to talk about the satellite cell. The satellite cells are flattened cells arranged around neuronal cell body in the ganglia. If I make this picture bigger, you can see the cell body of neuron in the ganglia, and these are the flat cells which can and cover the neuron cell body. We call them satellite cell. The other name of satellite cell is amphicyte. Let me type it here for you. We call them amphicyte too. The satellite cells physically separate cell bodies from their surrounding interstitial fluid. They function to electrically insulate the cell body and regulate the continuous exchange of nutrient and waste product between neuron cell body and their environment. The other Gilial cell we have in peripheral nervous system is called Neuronemocite or Schwann cell. The Neuronemocite are elongated cells, they are flat cells, and they wrap around the axon and insulate the axon within the peripheral nervous system. And they can make myelin sheath through the process which we call it myelination. As with myelin sheath formed by oligodendrocyte in the central nervous system, this allows for faster propagation of action potential along the axon. The process of forming the myelin sheath by both neuronemocite and oligodendrocyte is described later for you. So, the neuronemocite can make myelin sheath around the axon in PNS, while the oligodendrocyte can make myelin sheath in the CNS. If you look at this picture, you can see the cell body and dendrite, and this long extension is the axon. And you can see the Schwann cell which wrap itself around the axon to insulate the axon. So, the Gilial cells, which we have in peripheral nervous system, are two. One of them is satellite cell, which electrically insulate the cell body of the neuron in the PNS and regulate nutrient and waste exchange for cell body in the ganglia. And the other one is called Neuronemocite. It can make myelin sheath around the axon and insulate the axon, and it can allow faster action potential propagation along the axon. Neoplasm resulting from unregulated cell growth, commonly known as tumors. And most of the times it occurs within the central nervous system. A tumor that originates within the organ where it's found is called primary tumor because most mature neurons cannot undergo mitotic division. Primary central nervous system tumor typically originate in supporting tissue within the brain or spinal cord. In the Gilial cells, which can do mitotic division. The meninges or which can protect our central nervous system can show these tumors too, and I will talk about it later for you. Gilial cell tumors, termed gliomas, may be either relatively benign and slow growing, or they can be malignant, and they can do metastasis and spread to different parts of the nervous system. So most of the times, the Gilial cells can originate the tumors because the neurons cannot do mitotic division to make tumor. The next part is about myelination. The myelination is the process by which part of an axon is wrapped with myelin or myelin sheath. Myelin is the insulating covering around the axon that consists of repeating concentric layers of plasma membrane of a glial cell. Myelination is completed by neuronemocite in the PNS and oligodendrocyte in central nervous system. Myelin mainly consists of the plasma membrane of these Gilial cells. So they contain large proportion of lipid and a smaller amount of protein. The high lipid content of myelin gives an axon a distinct glossy white appearance and effectively can insulate the axon. Okay, look at this picture. In this picture, you can see the process of myelination. Here we have the axon, and this is the Schwann cell. The Schwann cell starts to wrap around the axon, and you see that it wraps, and wraps. And then we can see the concentric layers of the plasma membrane of the Schwann cell, which can make myelin sheet for us. So this is the plasma membrane, actually, which we call it myelin sheet. And here you can see the cytoplasm and the nucleus of Schwann cell. This is the other picture, which can show you the myelination of the axon here. This is the peripheral nervous system, and here we have the Schwann cell or the Neuronemocite, which wrap around the axon. Between these Neuronemocite, you can see a space, which we don't have myelin sheet. These spaces are called Neurofibril Node, or we call it Ranvier's node. These nodes are very important for jumping off action potential, which I will talk about it later for you completely. But if I want to talk about action potential here, the action potential can jump, in these Ranvier's node. So the action potential doesn't need to move all the length of the axon. It can jump, and jump until it reaches itself to the last part of our axon. Myelination is different in the central nervous system and in the peripheral nervous system. In the central nervous system, the oligodendrocyte can make myelin sheet. And you need to know that each oligodendrocyte here can make myelin sheet around several axons. Oligodendrocytes myelinating portion of these different axons in this picture. Look at here, this is one oligodendrocyte and it can make myelin sheet around this axon and around this one. And you see that by each of these cell process, it can make myelin sheet in one part of the axon. So in multiple spots, it can make myelin sheet. When we talk about the central nervous system, you need to know that we don't have any neurolema or Schwann cell here. And between this myelin sheet, you can see neurofibril node or Ranvier's node, which can help jumping of the information and action potential in myelinated axon. But in the peripheral nervous system, let me bring the picture of peripheral nervous system. In the peripheral nervous system, you see that we have different Schwann cell which can wrap around one axon. And they can make myelin sheet for single axon. And if the axon is longer than 1 millimeter, it takes many neurolemmocytes to myelinate the entire axon. The axon in many of the nerves in our body have hundreds to thousands of neurolemmocytes along their entire length. The gap between these neurolemmocytes, as I told you, is called Neurofibril Node or Ranvier's node. And this is the other chair, which can show you the unmyelinated axon in the peripheral nervous system and in the central nervous system. When our axon is unmyelinated, it doesn't mean that it doesn't have neurolemmocytes. We have many unmyelinated axons in our peripheral nervous system which are associated with neurolemmocytes. And these neurolemmocytes can protect and support the axon. However, they don't wrap around the axon and they don't make myelin sheath around the axon. And here you can see that we have a depression for the neurolemmocyte, which the axon are located inside. And here you can see the other Schwanzer or neuronemocite. It contains different depression, and in each of these depression, one axon is located. We don't have wrapping of neurolemmocyte around the axon. And also in the central nervous system, unmyelinated axon not associated with oligodendrocyte. So it's important to know about it. In this picture, you can see the Australocite, many cell extensions which can make blood-brain barrier. You can see the microgiliocyte in this picture, or microgilial cells or macrophages of the CNS. And here you can see the Pandimal cell. They are simple cuboidal or simple columnar epithelium which line the cavities of our brain and the central canal of the spinal cord, and they contain cilia and the function is making cerebrospinal fluid. And here you can see the oligodendrocyte, which can make myelin sheath and multiple axon in multiple spots. Pay attention. Unmyelinated axon in the central nervous system are not associated with the oligodendrocyte. And here you can see the neuron in the peripheral nervous system. Around its cell body, you can see the satellite cell, and around the axon, you can see many Schwann cells. Between these Schwann cells, we have Neurofibril node or Ranvier's node. We have a disease which we call it multiple sclerosis or MS. The MS is progressive demyelination of neuron in the central nervous system, accompanied by the destruction of oligodendrocyte. MS is an autoimmune disorder or autoimmune disease because the body's immune cell mistake the oligodendrocyte as foreign body and attack them. As a result, the propagation of action potential is disrupted, leading to impaired sensory perception and motor coordination. Repeated inflammatory events at myelinated site cause scarring or sclerosis. And in time, some function is permanently lost. The disease usually affects young adults between the age of 18 to 40. It's 5 times more prevalent in individuals of European countries than African American. Among the typical symptoms are vision problem, muscle weakness and spasm, urinary tract infection, and bladder incontinence. The other problem is called Guillain-Barré syndrome. And in this problem, we have some inflammations which can lose myelin sheath, our peripheral nervous system. In this disease, in this problem, we have muscle weakness, which starts from the distal limbs and progressively it comes to the proximal part. And by some medication, we can treat it. Now you can answer this question. If a person has a brain tumor, is it more likely to have developed from neuron or glial cell, and why? If a person suffers from meningitis and inflammation of meningeal covering around the brain, which type of glial cell usually replicate in response to the infection? And which specific type of Gilial cell can form a myelin sheath in the PNS? And what is the function of myelin sheath? How does myelination of axon occur in peripheral nervous system? And the last part of this video talks about axon regeneration. And you will learn that which factors can influence regeneration of PNS axon, and then you can explain why axon regeneration in our central nervous system is limited. And then you can describe the event of Valerian degeneration and axon remote. PNS axon are very sensitive to cuts, crushing injuries, and other type of trauma. However, a damaged axon can regenerate if the cell body remains intact and a critical amount of neurolemma remain. So if the neuron cell body is intact and we have enough neurolemmocyte, we can have the regeneration. And regeneration success is more likely if the amount of damage is less extensive and if the distance between site of damage and a structure which is innervated is shorter. Now I want to talk about the different steps which we have at the time of regeneration. Look at this picture. I will talk about them step by step. First, step 1. The axon is served by some type of trauma. In the second step, the portion proximal to the trauma seals off by membrane fusion and swell. The swelling is a result of oxoplasmic flow from the neuron cell body to the axon. At the same time, the axon served from the cell body and the myelin sheath surrounding the axon break down. The process is called Valerian Degeneration. Macrophages migrate to this part and remove the debris through the phagocytosis. However, the neurolemma in the distal region remain. The neurolemma of the Schwann cell, they are remained here. In the third step, the neurolemma and the remaining endoneurium can form a tube which we call it regeneration tube. In the step 4, the axon begins to regenerate and remyelination occurs. The regeneration tube guides the axon support as it begins to grow rapidly through the regeneration tube at the rate of about 2 to 5 millimeters per each day. These occur under the influence of nerve growth factor released by the Neuronemocite. And in the last step, which is step 5, innervation is restored as the axon reestablishes functional contact with its original structure. The structure is either a receptor for the sensory neuron or region sensory perception, or an effector for motor neuron to regain control of a muscle or gland. So you see that the regeneration takes place if the cell body remains and this space which we have between the site of trauma and our effector organ is short. In this picture, again, you can see all of this process. This is a neuron which can innervate this muscle. As you see in this picture, we have a cut in this part. And when we have a cut, our axon, which is lower than the site of cut, is removed by the macrophages, but the neuronemocite are remain here and make regeneration tube. On the other side, the end of the axon becomes sealed, and the axon starts to grow and find its direction in the regeneration tube, and it can reach itself to the muscle which innervates. During the time when the muscle is not innervated by the axon, you see that the size of muscle become smaller, and you can see it in paralysis. When we have a paralyzed person, you see that the limb which is paralyzed has a less amount of muscle than the other limb. And sometimes axon cannot reach itself to the muscle, and you see that it ends here. It cannot find its way to go to the Valerian tube or regeneration tube. So the innervation of that muscle is completely gone. Potential regeneration of damaged neuron inside the central nervous system is very limited for several reasons. One, the oligodendrocytes don't release a nerve growth factor, and in fact, they actively inhibit axon growth by producing and secreting several growth inhibitory molecules. Second, the large number of axons crowded within the central nervous system tend to complicate this regrowth process. And finally, both astrocyte and connective tissue coverings may form some scar tissue that obstruct axon regrowth. Medical researchers are attempting to overcome these limitations to treat patients with spinal cord injuries. And now you can answer this question. What two primary factors determine the effectiveness of PNS axon regeneration? And how does the process of nerve regeneration occur in the peripheral nervous system? In the next video, I will talk about neurophysiology for you. Have a great day.